Topic 5 deals with pollution. In part 1, we'll be looking at how we can monitor pollution in both terrestrial and aquatic ecosystems. What we're going to look at today is how we can, what methods we can use to monitor pollution. So we'll be looking at direct methods and indirect methods. Specifically, we'll be looking at biochemical oxygen demand and measuring dissolved oxygen. We'll also look at another indirect method using a biotic index. So a direct method is when you measure the actual amount of pollutant, whether it's in the water, the air, or in the soil. For example, in the air we might measure the acidity of the rainwater, so that will tell us how much of a certain chemical is present, or if we directly measure a certain gas in the atmosphere. In water and soil, we can test for nitrates. We tested for nitrogen in our soil lab. We can test for organic matter and also heavy metal concentration. So that's measuring the actual amount of the pollution. So you're directly measuring the chemical. Indirect methods, you look at how another factor changes as a direct result of a pollutant. It could be an abiotic or biotic factor that changes. Dissolved oxygen is something that we can measure, measuring biological oxygen demand, and looking for the presence or absence of certain indicator species. So the difference between the two, the direct methods monitor the pollutant itself, while the indirect methods monitor the effect that that pollution has on other factors. All right, so some types of direct measurements. If you measure the temperature in several locations along a river, or if you measure at different times throughout the year, that might help you measure for thermal pollution. So your pollutant is the heat. You're measuring for that amount of heat. If we're looking for nutrient overload, we would measure the amount of nitrates, the amount of ammonia or chloride levels, and that can be in a body of water or in soil. Total suspended solids and total dissolved solids, that would tell us the types of sediments that are in a body of water. That can tell us what type of pollutants are entering that river, entering that stream, entering that lake. Measurements that are indirect, if we look at the presence or absence of indicator species using a biotic index, and then the one that we're going to do in class is measuring dissolved oxygen to determine biological oxygen demand. When we look at indicator species, certain species tend to appear with pollutants while others tend to disappear. For example, the types of species we might look at, the fish, the bottom fauna, which would be the plant life, algae, bacteria. If you have polluted water, one of the types of organisms that tend to appear are the rat-tailed maggot, as you can see in this picture here. So if this lovely creature is present in the body of water, chances are that that water has been exposed to some sort of pollutant. If you see lichens on the trees, this would be an example of the presence of an indicator species indicating that there is no pollutant. So if the lichens disappeared, that would indicate some sort of air pollution. Coliform bacteria are a type of bacteria that grow in the water when there is sewage present. So one of the things that water treatment plants do is they go out and they test water in various places around the city to make sure that there's no coliform bacteria growth. And one of the things they do to prevent this is by adding chlorine to the water. A biotic index is a measurement on a scale of 1 to 10 that measures the quality of an ecosystem based on the types of species and how many of each are living in that particular ecosystem. We're usually looking at macroinvertebrates. And what it does is it helps us get an idea of what the effect of a certain pollutant is on the biodiversity. So if you have a fairly low biotic index, if you look in the diagram here at the bottom, ranging from 0 to 3.75, that is excellent water quality. That means that you have very little chance of having organic pollution. You can see as those numbers get higher, as you get closer to a 10, the water quality is very poor. So chances are you have some sort of severe organic pollution. One of the ways that Biotic Index measurements are taken is by volunteers. Volunteers will take a questionnaire and they'll go out and they'll actually look for these macroinvertebrates along the shorelines, in the water, along tree roots or logs that are in streams or lakes, and they'll fill out a questionnaire, they'll count the number of animals, and then they'll give an index score. Usually these booklets come with diagrams to help them identify the types of organisms that they see and pretty extensive instructions. So anyone can volunteer to do this. So you could volunteer and turn in your results to help with the measurement of Biotic Index. The one that we are going to concern ourselves with in class is biological oxygen demand. The amount of dissolved oxygen that is required to break down organic matter is what it is that we're measuring. So aerobic decomposition requires oxygen. So if you have a lot of decomposing material, you're going to have a high biological oxygen demand, which means that if your dissolved oxygen levels are not high, they will quickly drop. So the more organic pollutant that you have in the water, so that could be sewage runoff, for example, you're going to have more oxygen-consuming microbes breaking down that organic pollutant. So the demand for dissolved oxygen would be higher. In addition to a sewage, another example would be plant matter near sawmills, where a lot of the pulp is put back into the water. Animal feedlots would be an example of animal waste, like our lovely hog farms in eastern North Carolina. But what happens when these organic organisms break down, the dissolved oxygen is depleted, which affects the other organisms in that particular ecosystem. Now, if you have running water, flowing streams tend to recover pretty quickly. As long as there's still plenty of water and it's moving along, it's going to naturally pick up oxygen from the air. One of the problems is when too much sewage, or if you're also using that as a water source. For example, in developing countries, they take about 95% of their sewage and dump it directly into rivers and streams. Well, these rivers and streams cannot process that waste in the amount of time needed. So what happens is you get what is called an oxygen sag curve. So when you have this sag or depletion of dissolved oxygen, then certain organisms in that area are going to be depleted. So one of the ways that we can measure this is to take what is called a 5-day BOD test, or BOD5 for short. So you'll measure the dissolved oxygen on day 1, and then you'll go out and measure it again on day 5. You'll keep your sample in the dark during those 5 days, and that ensures that respiration is going on and not photosynthesis, that no oxygen is being added to the water. So then you would measure how much dissolved oxygen has been used, and that can help you determine what the demand for that oxygen was. Now, the oxygen sag curve. All right, so right here, this is a clean zone. This is a normal level of dissolved oxygen. At the point of discharge, so let's say it's sewage, into the river or stream, then the biological oxygen demand is going to go up. That's represented by our blue line. When that demand goes up, the dissolved oxygen goes down as that organic waste is being decomposed. Now you can see it disperses as we get further and further away from that point of discharge, and then we get into another clean zone. Now if you look closer in this diagram at the types of organisms, those that can live in lower levels of dissolved oxygen are what we call trash fish, like carp, catfish, whereas our normal clean water organisms require a lot higher levels of dissolved oxygen. So we can really determine what water quality is not only by levels of dissolved oxygen, but what types of organisms we see there. All right, so looking at this diagram, this compares dissolved oxygen concentrations in water across the world. So in blue was the 1980s, in orange is the 90s, and purple is the 2000s. So you can see how that has changed, and those dissolved oxygen levels once again indicate poor water quality.